morning everyone, I am Teacher Jude Michael Imperial, your teacher in science, and we'll discuss about the cellular basis of life, microscopy, different cell types, and techniques and methods in studying cells. But before that, kindly stand up and pick up the pieces of paper in front of you, specifically those crumpled paper. If it's all clean, may I call on Mr. Rebamonte for our short prayer. In the name of the Father, the Son, the Holy Spirit. Amen. Thank you, Mr. Rebamonte, for our short prayer. Is anyone absent today, Miss Cuevas? So you are all here. Yesterday, I have discussed to you and recalled about the biochemistry of the cell. Okay, for a cup of Nissan noodles, who can give me the definition of a cell? Mr. Mercadejas. Very good. Cells are the basic building blocks of all living things. The human body is composed of trillions of cells. Cells have many parts, each with a different function. Some of these parts, called organelles, are a specialized structure that perform certain tasks within the cell. For a special mamon, who can give me the 4 building blocks of the cell in its larger units or the monomers? Mr. Micosa. Excellent. The building blocks of a cell are carbohydrates, lipids, proteins, and nucleic acids. The monomers, on the other hand, are monosaccharides, amino acids, fatty acids, and nucleotides, respectively. Okay? During the pandemic, I know most of you have been in a long time staying at home because traveling have been disrupted by the COVID-19. Therefore, I have provided here a presentation in which you can at least feel that you are in those museums seeing faces that must be remembered when biology is concerned. But the twist is, you need to guess their names in this gibberish format. You know what a gibberish game is, right? It is a puzzle of words or emojis that sounds what it is imposing to. Okay, I'll give you an example. Who can guess it? Mr. Enrile? Correct. This is Rodrigo Duterte, our president today. Okay, class, get it? Okay. With gibberish number 1. Miss Nakata? Very good. This is Robert Hooke, who first coined or first used the word cell. Okay, next one. Mr. De La Cruz. Correct. This is Anthony van Leeuwenhoek, who invented the first microscope. Okay, for the third gibberish, Miss Lozano? Excellent. This is Matthias Schleiden. He is a German who co-authored the cell theory with this following two. Okay. Mr. Almeida? Alright. He is Theodor Schwann that expanded the cell theory to animals. And the last one is Miss Mansueto. Very good. This is Robert Virchow. He is known as the father of modern cellular pathology. This person, which are mostly scientists, developed and studied the cell theory. It is a theory stating that cells are the basic structural, functional, and organizational units of both single-celled and multicellular organisms. Cells divide and pass on hereditary information, and energy flows within the cells. For simpler version, the cell theory states that all organisms are composed of one or more cells. The cell is the structural unit of life, and the cells arise from pre-existing cells by division. But how can we picture out what are cells? here comes the microscope invented by Anthony Leeuwenhoek. I almost forgot. I have assigned you to print or have a cutted out picture of microscope, light or electron, right? We'll use that in the second activity. After I hand you these worksheets, kindly list 5 parts of microscope, identify it on the picture of the microscope you have printed, and give its functions. Okay. I have this for example. You cannot include this one on your worksheets. And here it is. This encircled part of the microscope is called the eyepiece or ocular. It magnifies the primary image produced by the objective. Okay, now, start with your worksheet, and if you are already finished, pass it to me and we will identify the parts of the microscope. Okay, time is up. Let us now give the function of each parts. Number 1 is arm. It connects to the base and supports the microscope head. So how you will handle a microscope? Grasp on the arm and hold the bottom or its base like this. Okay, for number 2, it is revolving nose piece, also called as turret. This part holds 2 or more objective lenses and can be rotated to easily change power. Number 3 are the objective lenses, which relays a real image of the object to the eyepiece. Next is rock stop. This is an adjustment that determines how close the objective lens can get to the slide. Okay. Next are the stage clips that holds the slide in place. It is attached on the stage, which on the other hand is a place for observation. Next is the condenser that serves to gather wave fronts from the microscope light source and concentrate them into a cone of light that illuminates the specimen with uniform intensity over the entire view field. For illuminator, it is to provide even, high-intensity light at the place of the field aperture so that light can travel through the condenser to the specimen. There are also 2 types of focus in the microscope. This one is coarse focus, which allows for quick focusing by moving the objective lens or stage up and down. And fine focus, to sharpen the focus quality of the image after it has been brought into the focus with the coarse focus knob. Lastly, the base, which supports the illuminator. Okay, sir, you mentioned earlier that there are 2 types of microscope. Yes, there are light and electron microscope. For light microscope, visible light is passed through the specimen and then through the glass lenses. Okay, for electron microscope, it focuses on a beam of electrons through the specimen onto its surface. Okay, next slide. The parameters in microscopy are magnification, contrast, and resolution. For magnification, it can magnify effectively to about 1,000 times than the actual size of the object. For contrast, it accentuates differences in parts of the sample. And for resolution, it measures the clarity of the image. Okay. With these microscopes, we can now see the properties and identify its fundamental types of the cell. For this, I will group you into 3 and you will watch a video about cells and its fundamental types. After that, you need to discuss the differences and similarities of prokaryotic and eukaryotic cells in front of the class. Group 1 is for prokaryotic, 2 is for eukaryotic, and 3 for the similarities. Okay, now, starts working with your groups. Yes, Miss Pelayo, what's your question? Okay, light microscope is smaller than electron microscope. Electron microscope is not usually used in the school laboratories.
Prokaryotic and eukaryotic cells are the building blocks of living things. Eukaryotic cells contain a nucleus and organelles bound by plasma membranes. Prokaryotic cells do not have a membrane-bound nucleus or organelles. The most obvious difference between the two types of cells is that prokaryotes have no nuclei, but there are really 5 major differences. 1. No prokaryotic cell has a nucleus. Every eukaryotic cell has a nucleus. 2. Prokaryotic cells have no mitochondria. Nearly every eukaryotic cell has mitochondria. 3. Prokaryotic cells have no organelles enclosed in plasma membranes. Every eukaryotic cell has a nucleus and organelles, each enclosed in plasma membranes. 4. Prokaryotic cells have circular strands of DNA. Eukaryotic cells have multiple molecules of double-stranded, linear DNA. 5. Prokaryotic cells are extremely small, much smaller than eukaryotic cells. A typical prokaryotic cell is of a size ranging from 0.1 microns to 5.0 microns. 1 micron or micrometer is 1/1000th of a millimeter, or 1/millionth of a meter. Anywhere from 200 to 10,000 prokaryotic cells could fit on the head of a pin. Their small size makes prokaryotic cells just 1/2 to 1,000th the size of a eukaryotic cell, which is typically between 10 and 100 micrometers. For all their differences, prokaryote and eukaryote have a few similarities too. Both types of cells carry on all the necessary functions of life. However, they do these things in different ways. Both cells carry DNA and rDNA. Both have vesicles. Both prokaryotes and eukaryotes may be single-celled organisms. Amoebas, paramecia, and yeast are all single-cell eukaryotes. Both have vacuoles, storage units for food and liquid. An organism with prokaryotic cells is a prokaryote. The 3 domains of life, eukaryota, bacteria, and archaea, include 2 branches that are prokaryotes. Bacteria, the first prokaryotes, discovered in 1676. Bacteria have bacterial RNA, no nuclear membrane, and cell membranes. Archaea, single-cell organisms. They have no nuclear membrane and share some qualities with bacteria, but are set apart from bacteria by their unique rDNA and ether-linked lipids in their cell membranes. An organism with eukaryotic cells is a eukaryote. Fungi, plants, protista, and all animals, including humans, are eukaryotes. We are all built with eukaryotic cells. Eukaryotic cells have nuclei and organelles, which immediately sets them apart from prokaryotic cells. The organelles in eukaryotic cells act as tiny membrane-bound compartments, performing all the functions of life in the cell: energy acquisition and transfer, digestion, waste management, reproduction, and cellular respiration. Eukaryotes can be single-celled organisms like protozoa or paramecia, or multicellular organisms like you, a tree, or an elephant.
Thank you for the presentation, Group 1, 2, and 3. Now, let us discuss first the basic properties of the cells that have been presented also in the video. Okay. It discussed that cells could grow and reproduce in culture and extended period, highly complex and organized, and possess a genetic program and the means to use it. In addition, cells are capable of producing more of themselves, carry out variety of chemical reaction and mechanical activities, and it can acquire and utilize energy to respond to stimuli. Moreover, cells are capable of self-regulation and evolution. Okay. For the 2 fundamental types of cells, we have prokaryotic and eukaryotic. It is distinguished by their size and types of organelles. Prokaryotes includes bacteria and archaea, while eukaryotes includes protists, animals, plants, and fungi, even humans. Prokaryotic cells, which are bacteria and archaea, have a simpler internal structure in which membrane-bound organelles are absent. In this type of cell, coupled transcription is direct to translation. Energy conservation reaction happens in the cytoplasmic membrane. Furthermore, DNA is placed in the nucleoid region, while eukaryotic cells, which are in the protists, animals, plants, and fungi, houses their DNA inside the nucleus, typically larger and complex. Here, DNA replication and transcription occur in the nucleus while translation occurs in the cytoplasm. Lastly, eukaryotes have membrane-bound organelles. The characteristics that distinguish the two cell types are the cytoplasm, cellular reproduction, locomotion, complexity, and genetic material. The common features of the two cell types are plasma membrane of similar construction, genetic information encoded in the DNA, similar mechanisms of transcription and translation of a genetic information, shared metabolic pathways, and similar mechanism in cellular respiration and photosynthesis. The differences of the two cell types, which have been discussed by Group 1 and Group 2, are the cell division, which is in nucleus and cytoplasm, chromosome size and location, cytoplasmic organelles for respiration and photosynthesis, cytoskeletons and appendages, and its cell wall composition. Okay. It has also been discussed on the video, the three domain of life, which are bacteria, archaea, and eukarya. Imagine a tree, which is the root is a cell and a Venn diagram in it, but it is not overlapping with each other. That talks about the differences of bacteria to archaea to eukarya, but all of them are cells. Okay, if you cannot imagine that, here's what I'm talking about. They are having different compositions, but they are all cells. This is the phylogenetic tree of life that is defined by the comparative rRNA gene sequencing. Okay, this revealed that studying cells are really expanding and progressing. And to help you with that, biologists suggested methods in studying cells through being knowledgeable enough about microbial diversity and prokaryotic diversity. Okay. Microbial diversity discusses that evolutionary facts revealed by the phylogenetic tree of life are 1. All prokaryotes are not phylogenetically closely related, and archaea are more closely related to eukarya than to bacteria. While prokaryotic diversity states that prokaryotes are identified and classified based on the specific DNA sequences, and prokaryotes are more diverse and numerous. Sir, how can we distinguish or identify prokaryotes? So here it is. It can be identified through phenotypic characterization, which are biochemical tests, G-staining, and other morphological characterization and molecular characterization, which is 16S rRNA gene analysis. Okay, again, what are the biologists' suggested methods that needs to be understood prior to studying cells? Mr. Cortez? Very good. It is microbial diversity, and 2 is prokaryotic diversity. Now, let us have a short quiz. Don't be afraid, it is just 5-item quiz on what we have discussed today. Get 1/4 sheet of pad paper and make sure that it has your name and your section. Everyone's ready? Okay, for number 1. It is an optical instrument used for viewing very small objects such as mineral samples or animal or plant cells, typically magnified several hundred times. It is an optical instrument used for viewing very small objects such as mineral samples or animals or plant cells, typically magnified several hundred times. Number 2, who are the 3 credited biology scientists for their works of cell theory? Meaning you need to list down the 3 biologists' full name, and wrong spelling is wrong. Number 3, what are the 3 domains of phylogenetic tree of life that is defined comparatively by rRNA gene sequencing? And number 4, what are the 2 types of a microscope? What are the 2 types of a microscope? And for number 5, it consists of a cytoplasm enclosed within a membrane which contains many biomolecules such as proteins and nucleic acids. Okay, if you're finished, group collector, kindly collect the papers of your group mates and exchange it to the other groups. Group 1 to Group 2, Group 2 to Group 3, and Group 3 to Group 1. Okay. Now let us answer the items. For number 1, it is an optical instrument used for viewing small objects, and that is a very good microscope. Okay. For number 2, the 3 biologists that authored the cell theory are, yes, Theodor Schwann, Matthias Leiden, and Robert Virchow. Okay, these are the spellings. Okay, number 3, what are the 3 domains of phylogenetic tree of life? It is bacteria, archaea, and eukarya. For number 4, the 2 types of microscopes are light and electron. And for the last number, it consists of a cytoplasm enclosed in a membrane is a very good, it is a cell. Write corrected by on the papers and encircle the scores. Give it back to the owner for clarification. And if it's none, kindly pass it to me. For your assignment to be submitted on Friday, I want you to make a brochure presenting differences of domain bacteria, archaea, and eukarya. Printed, or if it's handwritten, make sure that your penmanship is easy to read. You know that I always put attention to details, that's why I want you to be creative. For the criteria, 30 points for the information discussed, 20 points for the creativity, 15 points for grammar and composition, as well as the sources, total of 80 points. Sir, why are you always want to include grammar and composition and sources in your criteria? Okay, not because I am teaching science subject means that I will disregard other disciplines. I want you to be research-wise and adept on writing, which is also the essence of studying science and biology. By the way, your quiz today marks 10 points, okay? Oh. Any questions? Clarifications, violent reactions? If there's none, I will leave you with a quote from John Wooden that says, "Do not let what you cannot do interfere what you can do." Goodbye and thank you, class. Have a gentle break and God bless you all.